for the okay from, good, all right. Well, thank you all for coming. Very important day. We have Lydia here today. That's why it's such an important.
Sure.
Sure.
That's why I look like I'm about to cry. It's totally fine.
Besides the end of the world. So I'm going to give you a very brief introduction to Lydia, and then she'll take over. So Lydia Baker, well, they are our new colleagues next door here from CSUMB. We're very excited to have you. And remember, the open house, we had also one way to get to know you better. So they earned their PhD in oceanography from the University of Hawaii, Manoa, and focusing on diatom-associated bacteria. They completed postdoctoral research at Oregon State and Cornell, studying microbial interaction and symbiosis in anglefish and coral, respectively. And, well, currently, Dr. Baker is an assistant professor at California State University, Monterey Bay, where their research covers microbial ecology, symbiont evolution, and their impact on marine ecosystem. And I just learned that for the last 3 years, you were at Miami, University of Miami.
As a lecturer.
I'd like to have you here. So the title of their talk is Marine Bacterial Symbionts Challenging Evolutionary Norms and Informing Conservation. All right, thank you.
Thank you. Thank you all for coming. I'm going to hope this is as distracting as possible. I'm going to go to my happy place, which is science, so hopefully it is for you. This is going to be kind of a more highly technical talk than I usually give, because I figure I'm speaking to mostly grad students or people who are interested in this like highly, it's a difficult field to talk about because it's not an easily accessible one. There's like various levels of understanding and interaction, so I'm going to do my best to present some of the more difficult concepts along with the cool outcomes. So, my interests kind of vary. I've worked on anglerfish, I've worked on coral, I've worked on bacteria, mostly of course. I've worked on diatoms. So you might ask, what do all these things have in common? A lot of their life and livelihood depends on bacterial interactions. So that means that I've switched a lot of times with like the usual charismatic macrofauna that people kind of build their whole careers on. I just keep shifting. But as a consequence of knowing how to study bacteria, and especially how to study bacterial genomics and genetics, I'm able to work with all of these different species. And also, if there's anybody who works on any of that kind of larger cool stuff, and you have a cool question or you have an idea for a cool question, like give me some data. I will make it do cool stuff, guaranteed. So what is this cool data stuff, right? Like, so what we can do now, what I didn't get to do when I was a grad student. A lot of the times when we worked as grad students, we'd have like these little tiny pieces of DNA, and then we'd have to put these little tiny pieces of DNA into another bacteria, and then that bacteria would amplify it, and that's how we could tell stuff. Now we can take a whole anything. We can take tissue from an animal, we can take an environmental sample from the water out there, we can concentrate it down and isolate all of its DNA from it. So we can do two things with that. We can use amplicons, which are, again, those little tiny pieces of DNA, and we can find out, but those little tiny pieces of DNA, the way we get them is through polymerase chain reaction. So we can only really ask what certain groups are. So we don't have a universal way of finding all living organisms, unfortunately. So we have to ask questions about bacteria, or we have to ask questions about fungi, or we have to ask questions about other eukaryotes. But through that process, we can do a lot of good answering. So who's heard the term eDNA? So yes, eDNA is a big emerging field. eDNA is just what we've been doing when we study bacteria, except using different markers. So if you ever want to talk to someone who already knows all of the complexities of working with complex communities and little tiny pieces of DNA that you have to have a good database for, talk to a microbiologist, marine ecologist, whatever. We have lots of cool names and terms we shove in there. But the thing I really love the most is I always want more data. I want to know more stuff. I want it bigger. I want better. So what we can do is whole genome studies, and that's where we can actually look at every little tiny piece of DNA. And then we use cool computers to kind of like line up in the same way that if we went to Goodwill right now and there was like puzzles, right? Like say there's 5 copies of some puzzles and 3 copies of 2 puzzles and whatever, because it's goodwill, right? We shove those in a big, huge garbage bag. That's what whole genome sequencing is. It'll actually put those puzzles back together and use the duplicates of it to make sure that you have the right puzzles, because it's goodwill. They're probably missing a piece or 2, right? So that's what whole genome studies are about. But there's so much that you can do with whole genome data. Whole genome data can teach you not just about the identity of the organism that you're looking at, but its abundance in the environment. You can learn about how quickly it's replicating relative to other species. You can learn about the evolution of specific genes. You can look at differences between populations. So that's kind of like my bread and butter. So I wanted to just like shove in everything else that I haven't talked about too much yet into this talk. So I'm going to be talking about coral and shark and anglerfish. So it's going to feel like you're just going up and down in the water. So coral, I've worked a lot in coral-associated symbionts. I have a paper that's published in 2022 that I'd encourage you to check out. But I've also done an additional project that specifically looks at the coral nested ecosystem. Who's heard of the nested ecosystem concept before? Awesome. So a coral nested ecosystem is the idea, so coral itself is a symbiont, right? It's the interaction between the coral animal and the symbiodinium, right? So they interact together and they work as an ecosystem. However, if you consider like all of the other stuff that's living with a coral are covered with bacteria, coral are covered with fungi, coral are covered with microeukaryotes. We term that the holobiont. The holobiont really impacts the entire health and vitality of the coral. But that doesn't live in isolation. It lives in a larger ecosystem where there's interactions within the holobiont between that environment and the members of the holobiont. And this is why we call it the nested ecosystem. We're all on board still. So I'm just like, this is the fancy way of saying we sucked up coral mucus from right next to the coral, and then we looked at the bacteria in there. So we're not talking about the bacteria that are actually in the coral, but the bacteria and other things that are just kind of like close to it. So that picture is actually of my colleague, Dr. Cynthia Silvieri. She's at University of Miami. Her and her group got together and did this really extensive sampling of water along the leeward side of Curacao, focusing specifically on areas that are either human populated or not as human populated. And what they did was characterize the biome of these ecosystems. So if you want to check out that paper, that QR code is up there, and I can always turn back to it later if that's something that interests you. But she told me about this casually while we were having dinner, because I like being friends with other scientists, because that's super fun for me. And I was like, oh, did you look for bacteria? She's like, yeah, but we didn't find much. And I was like, mine, give me your data, please. And she was like, of course, why wouldn't I give you my data? So this is whole genome sequencing. So this is the kind of recommended best practices protocol as outlined by KBase. Who's heard of KBase? Awesome. So KBase is amazing because you literally can just upload any sequence, and there's tons of publicly available sequence in NCBI. So any question you have, KBase can probably find you. You can find some data, shove it in KBase, and KBase can process it, and you don't even need to learn how to code. You just need to learn how to push buttons in order. And that's the order that you push the buttons. So easy peasy, right? So you take the raw reads, you clean them up, and then you assemble them. After you've assembled them, you then, it gets binned. So it takes little pieces and says, oh, this is probably this thing. Oh, this is probably that thing. If I had to do this all by myself, like I'd probably cry. It's so hard. But we rely on these kind of tools to do it. They use what's called tetranucleotide sequences to differentiate, so just looking at the sliding scale of the different nucleotides. But again, any person could do this. I could probably teach my niece, who's 14, to go through this protocol. It's really simple. The important thing is to know kind of what you can get out of it, because at the end, you get these binned contigs, and they are going to be something. What you're going to get out of these specifically are bacteria that are the most prevalent bacteria in that environment. You need a certain number of copies of the bacteria for it actually to come out the other end. So you're not going to get the rare members using this protocol anyways. You could use this cool thing called kaiju, which I love the name too because I'm a big nerd. But kaiju can do it, can give you the kind of relative abundances of the more rare taxa. So these again are the sampling locations. They're they're lettered from A to L. And so these are how many bacteria I was able to find using that method I just outlined. We had some really common species in there. There's SAR-86, Acinetobacteria, an alpha proteobacteria that wasn't able to be identified any further, and a lot of Alteromonas. Alteromonas is very common in the ocean ecosystem. So this is fine, I guess. But I have a friend at K-Base. His name is Sean Jungbluth. He's also awesome and helped to build that protocol I was just showing you with all the little buttons. He pointed me to this paper because I actually went up for an interview at JGI. And he's like, you should talk about this in your interview. But it's actually the idea. This paper was revolutionary because what it says is instead of looking at every individual sample and putting it through that protocol, you should, if you have any kind of similarity in your samples, you should just like bin them all together and then do the protocol. Which to me was counterintuitive because of signal to noise ratio, which I think scientists are pretty aware what that is, right? So, but this showed conclusively that you'll actually get more bins with less contamination. I wanted to bring this to this audience too because there might be some students who are thinking about doing this or professors who are thinking about implementing this kind of protocol. The Joint Genomics Institutes, not even things that are in the NCBI, but specifically are at the Joint Genomics Institute, there are currently 219,000 uncultivated genomes, 123,000 isolates, 42 metagenomes, and 9,000 metatranscriptomes. If we went ahead and re-looked at this data and binned them by ecoregions, then maybe we can get more. So what's an ecoregion? An ecoregion is just a defined area where there are similar environmental resources that are generally similar. So all of this data has lat long information. We can put it against these ecoregions and then retry again. And you're like, well, why would I go through all this trouble? Like, how do we know for sure? This is just one paper. So these are the results of my individual binnings, and those are my results of my cat binnings. That is the difference between finding 8 bacteria and finding 21 different bacteria. You can also note, so that number at the bottom is the percent of aligned reads versus the total read effort. You can see how much it increases. So we're able to capture a lot more of the diversity this way, which to me was just kind of revolutionary. And I just was wondering, I was thinking back to my old projects, like what more could I bin? Like maybe I can find some more stuff. So if I use like kind of my regular like suite of tools, so looking at the relative abundance of there's 7 bacteria that were very well put together. They were more than 95% complete with less than 2% contamination. You can see that there's differences in relative abundance right away with Alteromonas and Q. flava and Synedococcus being especially prevalent in the areas where there's not as many humans. Now, if we kind of break that down into what their capabilities are, you can kind of see that there are differences in their metabolic capabilities, which suggests that they're responding to these different signals in the water. And if we go even farther and we go ahead and align the reads to the functional annotations, we can see that, so this is when you are aligning the reads to environmental indicators. You can see that the Alteromonas was negatively correlated with structural density and the total watershed, but it was positively correlated to coral cover. And Q. flava, for example, has a different result as it's just differently distributed. So basically, I'm saying you can take something like read abundance and be able to tell from metadata what the relative prevalences can be predicted by. This means that we can go into an environment and kind of predict what bacteria should be there. And especially when you go into like what they're actually doing in the water, it better informs our understanding of the biogeochemistry of that system and possibly what things that could be exacerbating existing stressors or even bringing it down. So that was coral. Who's ready for sharks? Sharks. Everyone loves sharks. I am the grand niece of, like at least academically, to Forrest Rohai. Forrest Rohai, I said his name is a scientific word that he got named after. Forrest Rower was the person, sorry, my advisor named one of the bacteria after him. Aquaricketia rohorii was named after him. So, but Forrest Rower has this wonderful concept that he's helped to develop, talking about the importance of apex predators and ecological impacts. This is also work that was perpetuated by Dr. Cynthia Silvieri. But basically, in a healthy environment where we have our apex predators, they're intact, our apex predators eat smaller fish, and those smaller fish are not there, so they can't eat the even smaller fish that are usually the ones that eat algae and are herbivorous. And so what happens is we have this certain level of bacteria, and that certain level of bacteria is counteracted by the viruses that are there. So as a consequence, we have what is usually a 10-to-1 ratio of viruses to bacteria. That is considered normal. Now, if we go ahead and kill all our sharks, no one's there to eat the medium fish, and no one's there to eat the medium fish will eat the smaller fish. And as a consequence, we have more algae. Algae tends to, especially macroalgae, produces certain carbohydrates that increase bacterial growth, and other factors that basically kind of lead to there being less viruses in the water. And most of us like, ooh, less viruses in the water, that must be great, right? No. Bacteria in the water, if there's more if the ratio is off and the ratio is less than 5, you get what's called microbialization. You'll get too much bacteria in that ecosystem per everything. So sharks really are kind of the control, especially in tropical ecosystems, to the rest of the ecosystem health. You'll also notice that this is kind of the environment too that like less favors coral growth. So sharks equal good coral. This is one of Forrest's like big pushes right now. So I like making friends with scientists. So I was lucky enough that Dr. Catherine O'Donnell decided to be my friend, and she runs Field School Miami. Field School Miami is a very equitable and inclusive space where they study and take out, I think they work 7 days on, 3 days off, and they've been doing that for the past 4 years. So there's lots of good interaction with sharks. Students are taught how to do basic shark tagging and handling. But as her and I were having dinner one night, she was mentioning that she kept seeing these black marks on her sharks. And it turned out that those black marks on the sharks, which I didn't realize how much that rhymes, but that's fine, are flatworm infections. So we had, Dr. Seckel is interested in parasites of that ilk. And she's like, oh yeah, I'm collecting those for him, but I don't know if there's anything weird with the bacteria. Do you want to say anything with the bacteria? And I was like, yes, of course. So we actually went out to go sample our first shark, and we were developing methodologies. And I was like, well, I don't want to have to get a swab and throw away a swab every time. I think that would be wasteful. So I started out with a scalpel. So sharks don't have a lot of mucus on their skin. So when I was trying to sample like fresh shark skin with a scalpel, if anybody's done that, it's real dumb. I was sitting there for like minutes, like with this like nurse shark bent over, like, come on. But I went off and tried to get the little patch, because they usually have this patch. It's right behind their dorsal fin, is where the flatworm infection usually is. I went to go scrape it, and the denticles just came up with the scalpel. And Catherine immediately was like, oh no. Because sharks' denticles shouldn't just fall out. Like, it's the equivalent of like teeth falling out of your mouth. It's very rare. So I was like, okay, we have something here. So we went on and we sampled quite a few clean regions versus the infected wound areas to try to characterize the microbial communities of flatworm-associated mucus lesions. This work was just recently defended by my student, Veronica Lopez, and is done both with the help of Catherine McDonald, as well as my associate Stephanie Rosales at NOAA, which is where a lot of the molecular work was done. So the thing that's interesting is that we have bull sharks and black tips. So bull sharks kind of hang out by themselves. This is actually a really good documentation of black tips in an aggregate. So using drone footage, we've seen that these black tips have aggregate behavior. This specifically was at South Point Beach in Miami, Florida. So it's kind of interesting that these species, like we're seeing these wounds on only these 2 species, and one of them has an aggregation behavior and one of them doesn't. So that still kind of like has us in a head-scratching moment. We don't know why only these 2 species have them because there's no reason that we can tell. We are working with an associate, Amani, who's possibly going to look at the differences in the denticles between the species. But at this point, we're still kind of like, no idea why. Because there's, I think, like 25 or something species that they sample pretty regularly out in Miami. So jump right into the data. So this is all amplicon data, so just using that little 16S indicator for bacteria. We saw really quickly that even though we have less bull shark, there were just less bull shark that have those lesions. That the healthy and for both the bull shark and the black tip have very similar microbiomes. So what you're looking at there is a breakdown of the different phyla where they're colored different colors. So there are a couple of healthy shark that have bacteroidetes, but they mostly are being taken over by proteobacteria, which is that nice green color. However, when you add lesion to them, we get an instant increase in the bacteroidetes species, which for us was like, okay, they're different. The bacteria are different. This, of course, is like broad resolution. This would be like, look, there's trees over there. I don't know what kind of trees there are, but there's more trees over there than there are over there. First, just checking out from like the broad view, right? So if we do this really cool analysis, which is called ANCOM-BC, it will actually pick out what the actual differences are in the amplicon sequence variants. So this is basically considered to be at like either species or strain level differences between the 2 samples. So when we look at the difference between healthy and infected, the ones that are, so this is what the microbiome of an infected wound looks like. We see that they actually have a drop in their streptococcus and their amoebophilus, but we have increases in their aquamarina, the mycoplasma. But what was really surprising is that the tenacibaculum increased in all of them. So tenacibaculum is a common fish disease, but it's not been seen in shark before. So just to give you another view, so this is that same kind of like abundance plot. You can see just how much the tenacibaculum, mycoplasma, listrangia increases as a proportion of the microbiome as a consequence of an infection. So to us, this kind of led us to kind of wonder, is the tenacibaculum leading to the flatworm infection, or is the flatworm infection leading to tenacibaculum? We don't know that yet, but it seems likely that it's the flatworms leading to the tenacibaculum infection, which is called tenacibaculum. You do fish. Have you seen this before? Okay. I'll show some pictures of what it looks like in fish. We think it's basically the flatworm infection is leading to this kind of, it's a black spot disease in fish because we don't see it, we've never seen this bacteria infecting shark before. There's also, yeah, it is that. But what's interesting though is like, so shark people have not noticed it before, but like shark people aren't necessarily always looking for black spots on their shark because usually wounds in sharks heal really fast. It's only because of the diligence of Dr. Catherine McDonald that she was even able to pick up that there were these differences in the first place. And then she reached out to me as a friend to see if I'd be interested in studying what it is, right? So it's one of the examples I think of like how important it is to have cross-disciplinary communication and also to be friends with people just because they do cool stuff. Sometimes you get to play along. So this tenacibaculosis has only been found and only been documented in one captive sand tiger shark. Treatment with erythrofloxacin antibiotic was successful. So I was like, well, let's go do it. Let's go cover sharks in antibiotics. And everyone was like, slow your roll there. That's not what we do to sharks. Also, this antibiotic has to be applied more than once, and so we don't know. But basically, so this is occasionally and rarely we'll actually find that these infections are down the fin, that main fin, and turn into these little bumps, and then you'll actually get the caving out, which is really closely similar to what you see. So that picture down at the bottom is the alaska.gov picture of what this species, what the same disease looks like exhibited in bony fish. So a lot of the times when we look at fish diseases, similar to when we look at coral diseases, sometimes we're just like, oh, that's a blah, blah, blah disease because it does blah, blah, blah. So this is a black spot disease because it causes black spots. This is yellow banding disease because it causes yellow banding. But like, I think it's very useful to have the conversation to actually establish that that's what we're seeing and establish metrics for it so that we can track diseases like this through populations, because especially in wild populations, we don't know if it's going to turn into nothing and it's just like, oh, they're sick, or if it's going to like ruin whole breeding stock, and we won't know until the breeding stock is gone. So it's one reason I get excited about stuff. Another thing I'm really excited about, we're still talking about sharks, don't worry, the big words, but I'm really interested in Rickettsia. There was this really awesome paper out in 2022 that I actually had started writing a similar paper, but they got to it first. That's fine. Talking about the evolution of Rickettsia in seawater. And basically, if you look at all of the known Rickettsia species that we have, we see multiple evolutionary tracks into seawater and away from seawater and into seawater and away from seawater. We see it going in and out of terrestrial ecosystems, in and out of brackish waters. So Rickettsia is a bacteria that is only been found associated with a host. We've never seen it found kind of on its own. But it's really cool because a lot of them, some species of Rickettsia can jump from host to host, which is, I think that's cool. That takes so much work. You have to figure if you're like this little bacteria and you don't have that many genes and you're able to like, oh, I can like infect a puppy and like I can infect a human. Like that takes a lot of work. You have to think about it like how much more genes would you have to take on? But just to show my bona fides with it, Dr. Morris Emalali at Lawrence Livermore National Labs reached out to me to do their phylogenomics for this, so I was able to look at a newly discovered Rickettsia marine species that was associated with a host that I can't talk about yet, but that paper should be coming out early next year. It's been in review for a long time because of Nature Communications, et cetera, et cetera. But in continuation with sharks, I'm especially interested in Wolbachia. Wolbachia is a bacteria that's in the Rickettsia group that can cause feminization of the offspring. And in the previous papers that have looked at this, only about 2 to 10% of the different organisms that they thought, so take a step back. Wolbachia is usually found in insect species. So the insect equivalent in seawater is copepods, isopods, stuff like that. Previous studies that have looked at different species of copepods have found that only about 2 or 3 or 5 different copepods actually harbor this bacteria, and everything else it's not there. I found this unbelievable because 60% of all 60% of all insects have Wolbachia. Like it's been found with them. So I'm like, we're missing all of this diversity of Wolbachia in marine ecosystems. So Emily Yeager, who is Catherine's PhD student, and I went out and just randomly sampled the water outside of University of Miami, because it's like University of Miami's marine campus is like right on the water. And 5 of the 9 samples we took had Wolbachia in it, which was surprising. And so we then decided we're going to start sampling all of the copepod species that we pull off a shark. So we did that, and the thing that we noticed is with both our random samples that we took off of the campus and the ones that we got off of the shark, every sample was preserved in a high-quality preservative, DNA, RNA shield, and we got something like a 70% return rate for everything that we tried to amplify. So it's another lesson, like I use high quality things to store my DNA because that's just how you do it when you're doing any kind of like free living bacteria. Like you would never dare put that in ethanol. But a lot of people use ethanol to preserve DNA when you're working with a large sample because there's plenty of DNA in there and it can't all go bad, right? But when you're thinking about working with symbionts, it's worth it to take the time. Okay. Anglerfish. So I was really lucky. So this is like my foundational work, what really got me into marine symbionts in the first place. I was lucky to work with Dr. Tori Hendry at Cornell to look at how diverse bioluminescent symbionts are in deep sea anglerfish. So what you're seeing here is the only video that I've ever seen. I do try to keep up with Bruce's output. It's the only one that actually, it's the only video of an anglerfish that actually has an attached parasitic male. So in that same turducken of symbiosis, some anglerfish have parasitic males, but their light is lit with bacteria. So they have two symbioses going on at once because they're fat and sit like that. But anglerfish are the most speciose fish in the deep sea. They're they differentiated about the same time that the dinosaurs were around. And so we would figure that their bacteria probably diversified at the same time. And we should be able to see the kind of like diversified evolution while still maintaining the same kind of ability to perform bioluminescence for their host. Because a lot of symbionts have trends. So genome size is directly related to how closely you're associated with a host. This overlaps a lot with how we kind of think about codependent relationships, for example, right? The more that you rely on your host or your partner to do something, the less you're going to learn how to do that thing. Like if your host is or if your partner is a really great cook, you're probably going to stop cooking as much and you're going to forget how to do some of the cool dishes you did before. Same thing with bacteria. So basically, if you're going to be transmitted and you're never going to see the light of day, you're only going to keep the genes that you need to be able to like reproduce yourself and to serve your host. Everything else is just going to be wasted. There's, of course, more complex reasons for that. It's not like the bacteria are like, no, thank you, I don't need this anymore. It just becomes less likely for them to keep those genes. So we know that the Hawaiian bobtail squid basically expels its bacteria all the time. So its bacteria has to live in the natural environment, so its genome's pretty big. We know that the flashlight fish actually transmits their bacteria from fish to fish within its pod, but doesn't get it like from parent to offspring. They're just kind of getting it from they're getting what's called like pseudo. And therefore their genes are like pretty small. Their genomes are pretty small. But in stuff like insects and Wolbachia, their genomes are super tiny because they're just being transmitted from parent to offspring and parent to offspring. Similar with this, I should say anglerfish, we're in that like weird, we don't know. Seems like they shouldn't be in that little middle zone, but that's about the genome size, and I'll talk about that a little bit later. But another thing that we can do, again using this genome data, is look at the evolution of the host and look at the evolution of the symbiont. If they match, then they are likely co-diversifying and therefore likely being transmitted from parent to offspring. Super cool, easy peasy. I love doing trees. Trees are great. If you go ahead and look at those evolutionary characters and you just get this big mess, it's called a tanglegram, then it's probably not vertical transmission, and the only thing that it could be is horizontal. Now, horizontal can mean environmental or it could mean through a secondary host, but it's basically just not going from parent to offspring. But in the case of like the pseudo-vertical, we'll see some matching and some not. And there's really cool tools on R that you can use. You don't have to like do it just by eye. There's like actual statistics that go behind that. But like it's really easy when you see a big tangle, you're like, oh, that's tangly. Don't know why the colors are popping up later, but cool. So if we look at all the other bioluminescent bacteria that have ever been characterized, you can see again there's that Hawaiian bobtail squid is like right there. And they have a pretty well-sized genome. It's in the 4 megabase pairs range. If we look at anglerfish, they are in the 2 and 0.2 range. So again, just kind of like put everything in context and give it some real numbers. And then when I joined the lab, we were super lucky because the Deep End Consortia, which is out of Florida, was going into the Gulf of Mexico because there was that terrible spill. And they were there to try to perpetuate that there was some sort of outcome to these species, to these deep water species. However, we don't have any like starting point for this, so all they had was like the end point of this. But as a consequence, we got 5 different species of anglerfish from them, and then we were talking to Dr. Spencer Nyholm, and we're like, we're doing a cool project. He's like, do you want more anglerfish? And we're like, of course we do. So he gave us 4 more species too. Previously, Dr. Tori Hendry characterized the bacteria associated with these 2 species, the 2 most common species. There's Cryptoceras kuzii and Melanocetus johnsonii. I forget its scientific name. I'm bad. But we found that there was 2 different species in there. So if we had 9 different species, what would you figure that we'd find for the bacteria that was associated with them? Anybody want to guess? 9 species, we'd find how many bacteria? 9, right? That makes sense. That's what I wanted. That's what I came in with. To give you some background too, because I didn't know this, but there's this awesome book that's just about anglerfish. I've read it all. It's like a textbook. Anglerfish reproduce, and of course we've gotten this all from like cruise data and like small observations, but we think the most likely way that they reproduce is that the male and the female release egg and sperm into these big mucosal nets. These big mucosal nets float to the surface, and at the surface they become little jelly beans that have no lure. Those little tiny larval fish will swim around until we have no idea. If anybody has the like ear samples, I would love to talk to you. There's no data on how old they are. Don't tell, I have no idea. But at some point, that little jelly bean decides that it's time to go down and be a deep sea fish. And so it starts swimming, swimming, swimming. And you can see its little tiny lure starts to pop out. And they find that the like juvenile that has its darkness and it has its full lure is actually deeper than the full-grown adult will be found. Do people know how big anglerfish are too, by the way? Do you have samples here? How big are they? They're tiny. They're so cute. I love them. I have a tattoo of them, by the way. I'm trying not to show it off right now because I'm hot. I don't want to take my jacket off. Yeah, so they'll actually swim and be way deeper down than the full-grown adults will be. And so our colleagues went ahead and got samples of all of the different life, except for, of course, the jelly mat, and went and looked for the bacteria in them. And they found that no symbiont was found in anything that doesn't have the dark coloration or the full lure. So this pretty much should preclude there being any vertical transmission. This isn't also like super, I don't know, it's not super convincing to me. You're just like, oh, we sampled it and it's not there. Like saying something isn't there is harder than saying what the other probable mechanism is. So we were given the whole lure, the actual like whole lure of the animal, DNA sequenced it, and then pulled out the mitochondrial genes. I keep saying we, I did this. I don't know why we always do this to ourselves, but we pulled the mitochondrial sequences out. And what you can see here, the tree that you see is all the ones that are colored are the ones that were from this study. They're all the different genus that were studied in this study. They match up with the animals that were observed. And what you see there is all the other mitochondrial sequences that were available at NCBI in the time. So we actually got a really good distribution of the different species that we were looking at relative to the known characterized mitochondria from other species available. So then I found this. And I didn't tell my advisor for like a month and a half. I only found 2 different species of bacteria. So I'm like, I did something wrong. Oh no, don't tell her, don't tell her. You're gonna get fired. But then I finally did, and she's like, okay, so what does that mean? Well, as it turns out, one species is specific to Cryptoceras kuzii, and the other species is common to every other anglerfish that we found. So it means we have a tangogram, which means they are not being transmitted from parent to offspring. interestingly enough, even though, because we got those samples from Dr. Nyholm and from the Deep End Consortia, we actually got multiple samplings from different locations. And so they're not differentiating by location, and they're not differentiating by host. So they're probably just like, that bacteria is maybe floating around in the water for a long time. It's also complicated because anglerfish in the Gulf of Mexico should eventually reach the Atlantic via thermohaline circulation. So it's kind of hard to place, and since we don't know how old they are. But basically, there's just no way. So even the one that's specific to only a single anglerfish host are not statistically likely to be vertically transmitted either. So they're just, these bacteria are living in the water for long periods of time. So one thing to note, one reason why their genomes are so big is because they're full of transposons and pseudogens. So the majority of their genome, not majority, there's just a big part of their genome that's just transposons. There seems to be some transposon families that are specific to each species of bacteria, but they're not specific to species of anglerfish, and they're not specific to the collection site. So even tracing the evolution of the transposon mediation through the species found that we didn't see any trends either. So the trees I was showing you before was a mitochondrial tree and a, genomic tree. So genomes using, I think, 150 different genes concatenated with each other. So it's a very strong tree. I also made a tree using single nucleotide polymorphisms, and it looked the same. So yeah, no, those are solid. This is some solid phylogenies, which I'm a big fan of. So this is the paper that resulted from our colleagues that were looking at juveniles and adults, just to show the data, the 16S data, and basically that purple species is the bacteria that we found that lights the lure, and you can see that it's missing from all the juvenile species. I do want to highlight, and it's always not good to say things about people, but I think this is a really good case. The initial analysis, when it was presented to me, was done with CHIME 1, and this was back in 2019. Their initial conclusions were that the juveniles had that bacteria with them the whole time because they found things that were in the Vibrionaceae category that were uncharacterized, and they were like, we're done. Green Genes is an older database, and CHIME does what's called clustering, and both of those things can lead to very spurious results, so I had to end up helping them redo the analysis and rewrite the whole paper because using the technology they were using, it was just wrong. Another thing that I did to help to substantiate this is I actually looked for the symbiont ASV in the water, and additionally I created a primer set to specifically look for those bacteria in the water and found similar results. I used that primer, and it was focused around a motility gene that was unique to this species, and motility in general is pretty low in bacterial populations in the deep sea, so lots of different double checking to make sure it's true. But basically we mostly found that our bacteria was found in the mesopelagic. So that bacteria is out there, but I keep forgetting to mention, the bacteria has a crud metabolism. It can only eat sugar. That's all it can do. It can't eat anything else. It can't metabolize any other form of carbohydrate. And the deep sea isn't a candy shop. It's mostly like used up humic substances and non-digestible carbons. So probably the bacteria are swimming from anglerfish to anglerfish using a little storage molecule that they have in them, which we can see through microscopy. But it's kind of revolutionary to think that like these bacteria are going between fish when they're so far from each other. It's not like you see schools of anglerfish. They're usually pretty lonely. So yeah, we're continuing this research actually thanks to one of the animals that was donated by Bruce Robeson, and the Mambari. We were able to get a melanocetus collected from California, and so that what you're seeing there on the left is single nucleotide polymorphism analysis of the difference between, so the red and the blue are Atlantic and Gulf of Mexico, respectively, and that yellow one is the Pacific. And you'd think they'd be more different. These species should be separated out by like a 500 years of evolution, and I don't think the animals are 500 years old, but the bacteria is not changing by different bodies of water, which I think is kind of surprising. But I'm still in process with that work with Dr. Tori Hendry. So take homes. Also, I love that video of Betty. She's great. So next generation sequencing is expanding our understanding of ecology and evolution. I didn't properly stress, but the finding with anglerfish was the first time we found a bacteria that has a reduced genome actually going from animal to animal as a symbiont. And I found the same thing in coral, which is just weird. But bioinformatics can expand the range of questions we ask and can make it accessible to students with minimal resources and training. And if you ever want to play on something, let me know. I love data. I'll play with you with data all day long. There's lots of usable data out there. Go into NCBI today, look up your species of interest, see what kind of things exist in the SRA, or the Sequence Read Archive. You can ask a lot of questions, or you can do meta-studies. It's just always there for you to play with. And marine ecosystems likely exert unique evolutionary pressures that will reveal these novel interactions, and unless we go and look for them, we're never going to find them. So, yeah. I'd like to thank, I've had a lot of non-traditional funders, so it's mostly through collaboration with networks like the Rosenstiel School. They would provide certain amounts of funding per student that I graduated, and I've graduated 25 different master's students. I've also gotten help from field school with my associate. That's Dr. Rosales on the right and Catherine McDonald in the center there. Through working with NOAA and the Shark Research and Conservation, as well as Cornell, Deep Halo, Deep End, and Lawrence Livermore National Labs. And I'd also like to highlight everybody's name more formally in case you want to look up their work there. So any questions, I would love to answer them. Oh, for the suit, like for everyone else, of course.
Also from home. Is it working? Yeah. Okay, let's start.
Thanks a lot. There's a lot of cool research.
I tend to like, I don't know, I'm extra, I guess is what the kids call it nowadays.
I think it's just the right amount.
Thanks.
You made a point in the beginning emphasizing the importance of binning samples to reduce contamination and get a better estimate of diversity and stuff. How do you deal with like the trade-off between that on the one hand and like replication?
So I didn't stress it, but in the protocol I was just showing you, they have a cool program that's called DREP that exactly handles that issue, where it takes into account, so usually they recommend using 3 different binning protocol, and then putting the results of those 3 different binning protocol into DREP. And so that kind of helps to even it out. And actually recently, I put the results of DREP for individual samples through DREP again, and was able to find that very reliably I was working with like 2 very different species of Flavobacter before. I wasn't quite sure if they were actually different. But DREP does it at enough high quality that it can actually differentiate between strains, which I think is a pretty powerful tool. So that's the trade-off. I mean, you always have a chance that you're going to throw away data. I think that's what you're probably more concerned about.
Yeah. Cool. Thanks.
Thank you. So I'm curious about shark denticles. Were any of the, they were sloughing where the lesions were.
Yeah.
Were any of the samples taken either from the same shark in an infected versus uninfected area?
All of our samples were done that way.
Oh, they were all done that way.
Yeah.
So when it was the log fold change, it was from the same individual shark. Okay, that answers a lot of the question.
Yeah.
Awesome. Okay, cool.
We're probably gonna, so the way that my student originally did it was without repeated measures, but we're implementing that now and finding a similar trend. So That's as if we treated those two samples as two individual samples. But yeah, it's pretty obvious if you look at even just the stacked bar plots that there's huge differences. So yeah, but we're in process right now, and we're hoping to have that work published by early next year.
That's great. Thank you.
For the bioluminescent bacteria,
Yeah.
right? The last sort of slide you were suggesting that in different oceans there's kind of the same bacteria species are there.
Yeah.
My question is, for other bioluminescent organisms, other fishes and things, do they have their different species of bacteria, or are they all kind of sharing these same bioluminescent species that are just floating around in this pool, and they just all grab them and use the same ones?
So that's a really good question. So there has been at least strain level differentiation that's been documented for flashlight fish that mostly goes along, because the flashlight fish are mostly found in the Philippines area, and there's that like line of, I think it's called the line of Henle or something like that, that's basically a big divide that they see in terrestrial ecosystems along that line, but there's actually a big divide that happens in, Wallace's line, thank you. Wallace's line. So they're finding that the species or the strains differentiate along Wallace's line there. But if you're going to talk about anything that's a free-living bacteria, yeah, it's kind of more like, cool, this works, I'm going to take it. But we don't usually see. We see that like there's one bacteria shared by lots of animals rather than one, like the other way around, where like one animal can just choose whatever bacteria is like lying around and bioluminescent.
So like in the same area, anglerfish and flashlight fish and lanternfish, they would probably all have similar bioluminescent bacteria in there.
I mean, they're really separated out. Like flashlight fish are like near shore, and
I was thinking more like locally, like there's lots of species of lanternfish, for example.
yeah.
Right? Would they all have the same species of bioluminescent bacteria in there?
From my understanding, yes.
Okay.
But the one I'm most familiar with is someone who was at the Cal Academy of Science. Her name's Ali something. I forget her last name. Sorry. But she did a whole paper comparing the like I was talking about with the Wallace line to museum samples of the same fish, and are seeing kind of consistent species use across two different species of fish. But yeah, that's another thing where it's like, a lot of these are really cool, but one of the reasons why we know so much about bioluminescent bacteria associated with squid is a lot of this has like worked its way into biomedical research, so it's able to kind of thrive in that environment. And it's a little harder to do when you're like, I study nearshore flashlight fish, or I study deep sea anglerfish. So the funding is mostly derived from people's startup, unfortunately. But if Monterey Bay Aquarium ever brought anything in bioluminescent, I'm stealing it. And by that I mean asking them gently if they could extract some for me.
I have a question. So earlier you were talking about how the anglerfish, the juvenile ones, they don't have the bioluminescent bacteria. And then when they're adults, then they get them. So do you know, like, do you have an idea of how and when they acquire those bacteria?
That's a really good question. And so again, our model is the Hawaiian bobtail squid, and we know like extensively they have like cilia, and they have like little mechanisms to make sure they get the right bacteria. This is not true at all for anglerfish. They just have this like lure that has a little opening to it, so we don't know how they're selecting for it. We know that the bacteria is capable, or has a lot of genes that are associated with flagella, so the idea is that they're able to traverse long distances with their flagella. And if you know anything about how quickly bacteria can swim, it's kind of mind-blowing, like how small they are versus how fast they run. I think I've done, basically if I was gonna, by the size of the room, if this was a bacteria, it would already be over there by the time I described how big the bacteria was. Like, they're fast. So yeah, so we think the bacteria are swimming around, probably using some sort of detection mechanism, like chemical detection method, to get to the anglerfish, and then finding their way to the lure. But like the statistical likelihood of that is really low. But they also experience these like pretty steep bottlenecks. So basically, and they also, because they have a big open lure, that they're just kind of like foofing out bacteria all the time, so they could be seeding the water all the time too. So
Thank you.
yeah. We've never actually seen an anglerfish like push out its bioluminescent bacteria like a squid would do it, but that's one of the hypotheses.
That's cool. Now I'm picturing these little like bioluminescent puffs. I was thinking about the coral mucus. I study sponges, and
Nice.
so I'm super interested in the sponge loop.
Yeah.
And so I'm curious about kind of the communities that are forming in the coral mucus. I mean, are they forming in the mucus? Because that was the sample, right? Was sort of the
Yeah, like
mucus.
mucus in the water
community.
around the mucus, yeah.
Yeah. So is the idea that the bacterial communities that are there are growing in there and developing there, or can you tell if there's colonization happening, or selection from something like sponge feeding or ascidian feeding that would be affecting that nearby?
That's a really good question, and the thing that is we're starting to catch up with in microbiome sampling is time. So a lot of the times we have just enough money to go out and sample once or twice and then come back. Each one of the samples, when it's a whole genome sample, is now, thank goodness, like $180 per sample, which is so cheap compared to what it used to be. If anybody wants to talk about where to get, is Azenta. Go talk to Azenta, they're amazing. And you don't have to have, like, we used to have full plates of samples, so you'd have to be guaranteed that you're going to give them like $5,000 worth of stuff, right? But now you can just give them one sample and like do genome stuff with it, and they're like, cool, got it. So adding time to that lets us answer those questions, but that means when I go to a granting agency, I'm like, I don't want 300,000 or 500,000 or 1.5 million. I want 3 million, and they're like, what are you answering that's that great? So that's one of the issues. So there are lots of cool questions like that. But what's fun too is like the statistics to actually look at what is basically repeated measures over time is really complicated too. It like adds like a tricycle to already juggling fire, which I do, by the way. Yeah. If anybody else does poi and like wants someone to play with, let me know. Any other questions? Cool. Yeah.
So that concept you were describing about like differentiating between vertical and horizontal transmission by the topology of the host versus the symbiont, and you were saying like you get a tanglegram versus a linogram or whatever you call it, right? Like, so how is it that you would have a similar topology of a symbiont to its host? Like, I mean, I would imagine they would be following different evolutionary pathways and.
The thing is they are, but they aren't, right? So you kind of think about it the same way that we think about like when we have two fish species that have been separated by an artificial barrier, right? One of them is going to look different from the other one, right? It's the same thing. They're going to look different from each other in a way that should be about the same time scales of differences that accumulate, and that's all we're measuring. Not necessarily that they're doing it with their host, but the host is, is their ecosystem. Does that make sense?
Like branching at similar time points?
Yes.
Okay.
Yeah.
Huh.
Yeah, no, it's complex. And it's really interesting because when I first presented this research, I did it at an ocean sciences type meeting, and people are like, this is nonsense. Like, what are you talking about? Like, why are you just making those conclusions? And it's like, we have literally decades of this working out in terrestrial ecosystems in our study of things like Wolbachia or study of other Rickettsia-like diseases. So, and not just them, but like lots of other bacteria go by the same thing. And there's actually really awesome work that's being done by, his name is John Stinton. I'm probably getting that name wrong. But there's also been studies that combine actually differentiation in the entire microbiome. If you do some kind of cool maths, you can actually see microbiomes differentiate along species lines, and we get co-diversification of microbiomes. Which makes sense, but also doesn't, because it's just all, you think of microbiomes as this like, more of a signature of like what you eat and where you are, not who you are, right? But it does really influence it. But they found similar trends in great apes and a lot of other terrestrial organisms, with the one glaring exception is that birds and bats actually have more similar microbiomes than anything else. And it makes no sense because they didn't, like, that's not how that tree branches. So they're still trying to figure out why birds and bats do weird stuff, and the only thing they can figure is altitude has a bigger effect than phylogeny, but that seems weird to me because we all go up in airplanes. Cool. Thank you all.